Well, good afternoon. I'm Diane Quinn from the Marine Science Center. This is our lecture series, The Future of Oceans. We're so happy that you're all here on this very wet day. Hope you like being close and warm. I'd like to begin by acknowledging that we work on the traditional lands of many Sklallam people, many Coast Salish people, particularly the Sklallam people, the Strong people. We respect and are grateful for this land and these waters and the people who have inhabited them since time immemorial and who continue to steward this place for future generations. I'd also like to acknowledge our very generous and gracious donors, the Darrow family, who have made, I didn't even have to say why, who help us keep this series accessible for everyone. And we are so appreciative of the fact that they think that adults might want to learn science too. So a couple of announcements. The first thing is our last lecture for this series will be on March 30th, and it will be at the Wheeler Theater next door, which is a little bit bigger than this. And the last thing I just want to say to everybody before I introduce our speaker is that the state parks, this park in particular, is getting ready to institute Discover Pass for the entire park. So for people who have been used to parking on the upper campus without a Discover Pass, starting March 1st, you will need a Discover Pass to park, which means that even to come to our lectures, you will need to have a parking pass. A couple of things. The Port Townsend Library has Discover Passes that you can check out. So if you don't have which is so cool. So if you don't have a Discover Pass, that's still no excuse. But we hope that you will not give up coming to the lectures because there's a parking fee. It will pertain to really everywhere on campus and every lot, everywhere, which we support because we want to keep our state park as healthy as it can be. So, yep, they're home. So today's lecture is just the same format as the others. How many people have been to other lectures this year? Yay, thank you. Okay, so after our speaker, we'll do Q&A for as much time as we can squish in, and I'll have a microphone and I'll try to, I don't know how I'm going to do it, but I will try to float above you to bring the microphone to you, and then our speaker can stay in place and answer questions. So with all of that said, today's speaker is Dr. Paul Yancey. For more than 40 years, he's been an ocean explorer. Would you love to be an ocean explorer? Yeah. Contributing to the cutting edge of what is known about life in the deepest parts of the ocean. His career-long work has focused on the physiological adaptations of marine animals to a variety of environmental factors in 3 extreme deep-sea habitats: hydrothermal vents, hydrocarbon seeps, and the tectonic trenches, habitats increasingly threatened by pollution, drilling, and mining. He's been on over 20 deep-sea expeditions around the world, including dives in the Alvin research submersible, which is the Woods Hole submersible, and the research vessel Falkor with Schmidt Oceans Institute. He and his work have been featured in filmed at sea documentaries like the one that's on Amazon Prime, NHK's Deep Ocean series, narrated by David Attenborough, and Expedition Deep Sea for the Discovery Plus channel. And he advised on BBC's Blue Planet 2, but he's also been interviewed in radio programs for NPR, CBC, BBC, and his list of publications and speaking engagements is very, very long. So we are very lucky. Paul has a bachelor's in science in biology from Caltech, but his PhD is in marine biology from Scripps Institution of Oceanography. And this is just a little cool thing that I'm just telling you guys right in the front row. In 1979, he won the Eckhart Dissertation Prize at Scripps Institution for Best Doctoral Thesis based on originality and significance of research and clarity of writing. And these are things that he has maintained all his life. I know. After postdoctoral work at St. Andrews in Scotland, he joined the faculty at Whitman College as a professor and the Carl E. Peterson Endowed Chair of Biology, now emeritus. Paul taught and mentored countless undergraduate and graduate students over 40 years, helping them to become the next generation of ocean researchers and champions for the future of oceans. In the free time that he's had over these 40 years, his summers and sabbaticals, he's been a visiting professor or a visiting researcher at institutions around the world, including the National Institutes of Health, applying his research findings to medical advancements, Oregon State University's Hatfield Marine Science Center, Stanford University's Hopkins Marine Station, Monterey Bay Aquarium Research Institute, University of Hawaii's Oceanography Department, and universities from Aberdeen to New Zealand. He has a lot of stories to tell, and we're very happy to have him here today, so please help me to welcome Dr. Paul Yancey.
Ooh, what an introduction. Thank you, Diane. Well, it's a real pleasure to be here, and I just retired a year ago and moved out here from Whitman, which is in Walla Walla. Anyway, I'm going to get right to it. By the way, if you can't read some little things like that in the back, don't worry about it. Those are for the YouTube, like copyright things and whatnot. I'll point out all the important stuff. But I thought I would start off, hopefully you've been watching this fly through the Mariana Trench, the world's deepest place. Talk about what we have to do to research the deep sea. So first of all, you're typically going to need a research vessel, and these are just some of the many I've been on, like the pressure drop, the Atlantis, and the Falcor. Some, like the Atlantis, are owned by the government, National Science Foundation, US. Falcor 2, this is Falcor 1. Falcor 2 is the world's most advanced ship. That's by Wendy and Eric Schmidt of Google. So theirs are much more advanced than the government ships. I was on the Falcor 1. And yeah, they all have stories to tell, but I've been on probably 15 different ships. They all have sonar. Nowadays, of course, we use multi-beam sonar that can give you these 3D plots. It's, these false colors you always see, right? So purple being the deepest. We still use things like old-fashioned technology, nets and traps. This is like a big fancy crab pot, basically. Landers. We also drop instruments over on cables, but landers are free falling. You drop them over the side. They have cameras and bait and so on. And there'll be a float attached. And maybe 24 hours later, you send an acoustic signal and the thing drops some weights and the float brings them back up. So those are landers, ROVs, you've all heard of them. I'm sure this is the Nereus from Woods Hole. And there are also autonomous ones. These are run by a cable from the ship. These run on their own on a computer program. They're more and more being run by AI. So maybe they'll take over the oceans, I don't know. This one, we learned the hard way why it's so hard to research the deep sea. The Nereus, on our last dive in the Kermadec Trench off New Zealand, imploded from the pressure. We lost it, so we didn't have it for the Mariana Trench, which was our next stop. We had landers. Anyway, that's the problem with deep sea research. You all have heard about imploding subs, I'm sure. And then we have DSV, sometimes called human occupied, but mostly deep submergence. The most famous one, of course, is the Alvin. The first version came online in 1964. It's US government owned. There's one of my students. And yeah, it's quite something. It's a top highlight of my life to go down in the Alvin. And One of the most amazing things is this is Victor Vescovo, a hedge fund manager, decided to build his own sub. And I got to go out with it. I didn't get to go in it, but I got to go out with it to the Antarctic. But it's called the limiting factor, holds 2 people. And the big difference is this can only go to 6,500 for the latest Alvin. This goes to 11,000, which is the Mariana Trench. So no government vessel except the new Chinese sub that just came out, but our government vessels can't go that deep. This private one can. Oh, and then finally there are cabled sensor array networks and so on being built. Permanent cables that run out. This is the University of Victoria's Ocean Networks Canada, runs out to the hydrothermal vents where I went. University of Washington runs another one just south of this. So those are different ways of studying the deep sea. All right, I'm going to move on to do an overview of deep sea habitats. So I'm going to have some kind of dense information at the beginning here to set the stage. And so I've got this generic diagram and a kind of a schematic of the deep sea. And remember grenadiers and sea pigs. This is from the University of Washington's cable network, but anyway, they'll come up. Sea pigs in particular. Anybody know what a sea pig is? It's a type of sea cucumber. All right, you'll see lots of those. First thing I want to point out is basically the deep is basically 95% of the ocean. And the reason for that is that this line here is 200 meters. That's about the level that photosynthesis occurs in the clearest water. And below that, there's no photosynthesis. So down here, no plants, no algae. And so we call that the deep sea. So that's 95%. And that's less than 10% explored. These are called the pelagic swimmer and floater zones. Epi, meso, bathy, abysso, and hadopelagic for the trenches. Less than 90% explored. Over here are the benthic or things that are on or just above the bottom. These are benthic animals. Subtidal, the bathyal, abyssal, and hadal for the trenches. And that's about 26% mapped as of last summer. But when I started, it was less than 10% mapped. The 3D sonar plots, well, the multi-beam are making inroads finally, but only 6% mapped. So this is the cutoff for the deep sea. Down here, this zone has some sun, but there's no photosynthesis, sometimes very low oxygen. Down here, all of this, no sunlight at all, colder and colder, pressure going up linearly. But there's plenty of oxygen down here for reasons I don't have time to get into, but that's another story. Okay, so I want to give a bit of history because I think this is important to set the stage. All of deep sea research goes back to the Challenger. This was the 1870s expedition the British sent out basically to find resources for their empire. And they went all over this red line here. They trawled down to 8,100 meters, and the Mariana Trench is 11,000, so remember that number. But they got the edge of the Mariana Trench, and so the deepest spot is named after them, the Challenger Deep. And they didn't know it was a trench, though, so they didn't know anything about trenches. They just got the edge of it, or, well, the upper end of it. And they kind of established what we all think of as deep sea nowadays, like anglerfish. They found all this stuff, vampire squid, anglerfish. You'll see all this stuff better later. But grenadiers and sea pigs on the bottom are the most common things. They established all this, and the food chain basically has been this way since the Challenger expedition, that everything starts up with sunlight, and then things die and sink, and animals and microbes consume it as it falls down. And the dogma has been that only a small amount reaches the bottom, and that's still true for the most part, but there's a big exception we'll get to. And so the animals, no herbivores, of course. Scavengers, detritivores, filter feeders eat different size of dead materials, and carnivores. So we'll get to all that. But that was established back here, 1870s. All right, and because their nets didn't catch very much down here for a century, till the 1970s, we thought deep sea life was pretty scarce. Okay, so they also discovered polymetallic nodules. Have you all heard about these? They're in the news a lot. Okay, we'll get to those. I have one up here if you haven't looked at it yet. Anyway, we'll come back to that, but they're, on this diagram, they're scattered around the abyssal plain, as we'll see here, the abyssal zone. Just a few more quick discoveries. 1933, the first seamounts were discovered. I'm not going to have time to talk about those. The trenches were not discovered till the Challenger 2 expedition in 1951. Deep trenches were all plotted out with the old-fashioned sonar. This is one of the extreme habitats I've worked a lot on. At the bottom of the Mariana Trench here, 36,000 feet, it's 1,100 atmospheres. That's about 100 elephants standing on your head. That's the weight of the water. So that's an extreme habitat. Now there's a myth that animals down there should implode or explode when they come up, but that doesn't happen at all, and we'll see why later. Basically, if you don't have a lung or a swim bladder, there's nothing to implode. The Nereus had cameras with air pockets in them, and we think one of those imploded. But if you don't have an air pocket, you're not going to implode or explode when you come up. Mid-ocean ridges were discovered by Marie Tharp, plotting out hundreds of sonar tracks. She found the rift valleys, which is where the vents would be found. Then plate tectonics came about, and a few other things is the Alvin was used to find the hydrothermal vents, extreme habitat from extreme temperatures and poisonous gas, and then the seeps up here, which I probably won't have time to talk about. Trenches here, vents are in the rift valleys that we'll get to, and I don't think I'll have time, like I said, for the seeps. But this is an area where there's huge amounts of frozen methane gas that various countries like Japan and China want to harvest, and there's delicate ecosystems on those, and I've worked on those as well. All right, that's a quick overview. So I also want to review where we're going. We're going to go to the rift valleys and the trenches mainly, and this is a review of plate tectonics. I'm sure you're all familiar with this since you live in an active area, but you all know the Earth's crust is broken up in all these different rigid plates, and they're moving. And some are spreading, of course. That's the light blues on here, mostly the mid-ocean ridges. And you can see the famous mid-Atlantic, of course, is the jigsaw puzzle of Africa and South America. So that's a spreading center. So they're spreading apart. Here, the plates are colliding, and we have one plate diving under the other, and you always get volcanoes. You get the heating up in a volcanic arc. So whether it's an island chain or the Cascades, you always get volcanoes. You don't always get a trench, as we'll see. And then there's transform faults. Those are all the dark purple. San Andreas is the famous one on land. They're all over the oceans. We're not going to deal with those, but that's the other movement like this. So you have spreading, colliding, and sliding, basically. Okay, so, and this is why the Mariana Trench is the deepest, because the plate has been spreading from way over here all the way over there for millions of years. So this is familiar to all of you, right, that live here, I hope. Yeah, so this is us right there, and here we are. So yeah, here's the Olympics, and here's the Juan de Fuca Ridge, and here's the spreading center. So we have this little plate here running over, dives under, melts, and you get the Cascade volcanoes. So we live here. For better or worse, we don't have a trench. And the reason is that the Olympics plugged it up, basically. So I don't have time to do geology for this, but basically the Olympics are ancient sea material, ocean material that got shoved up and pushed up, and the trench is gone. I think it's worth it. But because of that, I had to go to the Mariana and other places in the world, so there's no trench. All right. These are the ones I got to do. This is the Mariana over here by the Marianas Islands, Kermadec Trench, and the South Sandwich Trench down here. And then I did the vents and the Oregon seeps over here. So that's just to lay that out. Okay, so some cartoons. The little Alvin here can't shine enough light to get a picture of all of this. You can't get a big picture of it. So we have to have these diagrams. So I'll show you close-up pictures. But anyway, so here's a diagram of the Mariana Trench diving under, and you get these volcanic islands out here. And here's what the Juan de Fuca Ridge looks like. Here's the Rift Valley, where the two plates are splitting apart. This one that way, and one that way. All these lava formations, and here's the hydrothermal vents. Now this you can see. All you have to do is book a ticket to Iceland. So if you go to Iceland, the Mid-Atlantic Ridge comes right up out of the ocean, and there it is, and that's exactly this. That part of Iceland's North America, this part of Iceland is the Eurasian plate, and here's a hot spring. And if you go a little further north, which I did, you can find hydrothermal vents. Okay, that's a hydrothermal vent with a chimney. A vent is basically a continuous geyser. It's not intermittent. So those are in Iceland, and that's what's down here. Well, I mentioned this before. My fellow graduate student, Kathy Crane, I have to do this because she doesn't get proper credit. She was told that these are impossible in the deep sea, but she proved everybody wrong, and she's the one that discovered them because of this. She knew about this. But anyway, we'll get to that briefly. All right. So let me show you some of our real animals finally. This is where I started working, is on these sort of intermediate depth ones, these intermediate pelagic and the benthic ones. And you also, you catch them by nets, or sometimes you get some cool ones when you're in the Alvin or with a lander. This is the headless chicken monster, sea cucumber, went by one of our landers, and yeah, that was fun. The Dana squid, it's a very aggressive species, attacked the Alvin. It's like, I don't know what it's thinking. This one was really fun. The cockatoo squid came up and stared at us through the porthole. It was kind of like a bonding event or something. I don't know. It just stared and stared, and then it took off. This is part of a group called the glass squids. They're completely clear and most of their body.
What's the scale? Is it like this big?
That one was about, this is the sediment cores on the Alvin. I should have, the Alvin has laser calipers to measure it. As I recall, it's about this big. Okay, the two most common things that the Challenger found are grenadier, or sometimes called rat-tail fish, and sea pigs, sea cucumbers, so remember those. Joe and I did lots of work on these. Here's some of the other things I've worked on. This is an anglerfish that we had alive on the ship in the tank. That anglerfish is about this big, like, and it was sitting there snapping away on the ship in the tank. Lovely telescope fish, dragonfish, new eel pout species we discovered. Doesn't look much like an eel, so we nicknamed it Pudgy, but it was a new species. And here's some other fun deep sea things I've worked on. Giant isopods, your roly-polies from your garden. These are huge. Dumbo octopods, sleeper sharks. These are all benthic in the sense that they stay near the bottom. Cold water corals are all over seamounts. Anyway, there's lots of stuff I could go on, but you should know, if you don't already, is that grenadier are being caught and sold now because surface fishes are being depleted. So this is from a Russian website. They're fishing this giant grenadier like crazy, and they can't recover. These fish are sometimes 70, 80, 100 years old, and I don't know, it's kind of distressing. Pike's Market took grenadier off because it's not sustainable. They used to sell them at Pike Market. Okay, I'm going to just tell you briefly about what I do. I do a lot of different biochemical, physiological studies, but the main stuff I worked on are things called osmolytes, and the name means it controls osmosis. They're small molecules that basically keep cells from losing water, and you all know these two for sure, even though you might not. Taurine, I'm sure you've heard of because it's in energy drinks, right? It's the #1 ingredient in most energy drinks. There's lots of things you need it for in your body. I'm not going to get into that, but there's some in the hydrothermal vents. The one that you'll see later in the video is TMAO, trimethylamine oxide, which has a faint kind of somewhat pleasant fishy odor. It's what you smell in a fish market. And you know this for sure because you live here. When your Dungeness crab goes bad, it's because TMAO broke down into trimethylamine, which is literally rotten fish smell. So when you smell a bad Dungeness crab, it's my molecule. This is what I'm best known for is what does this thing do? All these marine animals have taurine and TMAO. TMAO, taurine, people knew what it had other functions, but TMAO was a bit of a mystery until I solved it. But all these osmolytes have other functions. They don't just help hold water. They have lots of functions that we've been researching over the years, and they're used in bioproducts and stuff because of that. But here we are smelling this really rotten delicacy in Iceland. What we discovered in a nutshell will be in the video later, but we found that this molecule protects fish from pressure. It keeps them from shrinking in a way that we haven't talked about yet. So we'll get to that. All right, so I'm going to move on to some habitats and the theme of human impacts that I'm going to be doing as we go along. And this one I haven't researched myself, so I'm going to give you a brief overview of that, and then we'll move to the trenches and the vents. But you've all heard of or a lot of you have heard about the polymetallic nodules, I'm sure, because they're going to be mined, and they're already starting. These things grow in rings over millions of years, and we don't know really how they form. There's still a lot of controversy about how they form, but they're full of metal oxides. And since the Challenger found the first ones in the deep sea, they've been found in all these red spots here. Hydrothermal vents we're going to come back to later, but since they were first discovered on the Galapagos Ridge, those have been found all over. So all the red dots, vents everywhere. Really important, as we'll see. The nodules have all these new ecosystems, well, not new, but new to us that people are discovering, like the Casper octopus is a new one, the Barbie pig, sea pig is a new one just discovered, 2021, and these are like 2020. All kinds of stuff growing on these nodules, and where you've all probably heard about them is they contain all these metals like cobalt and molybdenum and titanium and rare earths. What do we need rare earths for? Anybody remember? What's that?
Batteries and electronics.
Batteries, yeah. Rare earths are also all the colors in your phone pixels, your TV. The pixels you need rare earths for all those colors. So nodule and vent and even seamount mining is already underway. They've built these tractors, I'll show you one later, to harvest the hydrothermal vents and the nodules and even the sides of the crust of seamounts. And one of the big problems is when they suck these up, they have to get rid of the excess tailings, and those have been shown to kill fish, right? All this massive clouds of excess sediment. So that's stuff you all need to be aware of is going on. The first company to do this is probably going to be our neighbors. The metals company, their stock just doubled last week. Polymetallic nodules are the cleanest path toward electric vehicles. So this is Vancouver, right next door. They're all gearing up to it. They've already done some pilot studies. So this has already been done. It's just not commercially viable yet, but it probably will be soon. And I have a nodule up here. If you haven't looked at it, it's this one. And I also have a baby hydrothermal vent. So you should look at those. And if you look at the hydrothermal vent, you'll see it's lined with mineral crystals on the inside. So you can't look at them and say those are full of precious metals because you can't see them, but they are. They're full of all kinds of metals. Okay, China is also in a race with Canada. They're gearing up with new ships right now as we speak. And in the meantime, the United Nations is trying to regulate all this, and that's another talk in itself. Lots of opposition, protests because of the unique ecosystems. But anyway, you need to be aware that this is happening, and our neighbors may be the first to do it from Vancouver. Okay, so let's move to the trenches. And a little bit of history here. Back in the day, right after the trenches were discovered, there were a few things found down there with nets, like amphipods and sea pigs and a variety of worms and so on. And about halfway down a couple of trenches, like the Kermadec, they found this thing called a snailfish, and it was all mangled and beaten up in the net. And if you don't know snailfish, they're a group of species you find all over. Lots of different species. You can find them in tide pools out here. They're kind of a gooey, gelatinous set of species with a sucker disc. But this was one in the trench. And then that was just kind of forgotten after 1952. And there's a lot of other studies that I don't have time to go into, but I want to jump to the modern era, so to speak. The first landers were by one of my professors at Scripps, Bob Hessler, Philippine Trench. He found huge swarms of these amphipods, way more than anybody thought. And then my friend and colleague Alan Jameson in Scotland finally got cameras that could withstand the pressure of the deep by 2008. Before that, he couldn't do it. And he designed these landers, and this kind of blew everybody away. I'll show you videos of all this later in a few minutes, but this 1952 snailfish that had been almost forgotten, hundreds of them. They just came to his debate on his camera out of nowhere. New species he discovered in the Peru, Chile trench, made all the news media. And the amphipods, this is a nightmarish thing I'm going to show you here, but this is in the Peru, Chile trench, but we got the same thing I'll show you later. In the other trenches, amphipods. And this is a 24-hour time-lapse video that he did of, you'll see the bait go down on the lander and hit the seafloor. Okay, so that's the bait, it's going down to the seafloor. And here come the amphipods and a confused sea pig over here. And these are the amphipods, and here comes the poor sea pig. Sea pig doesn't know what to do. Says, let me in, let me in. Anyway, in 24 hours, the bones are stripped bare. So this is where you want to dump a body, I guess. But and then the lander comes back to the ship with its float, and it's stripped clean. So Alan and I got together right after that. I'm not going to go through all this in detail here, but we did, Jeff Drazen from Hawaii, and me and Alan and Tim Shank from Woods Hole, we got together, created the Hades, the Hadal Ecosystem Studies Project, to do the Kermadec and Mariana Trenches. And we recruited a whole bunch of other people you can see down here. I don't have time to go through everybody, but I list them all at the end for the YouTube. But anyway, we got together and created this trench project, Hadal Ecosystem Studies, Hades. And this is our plan here. So this is the false color plot of the part of the Mariana Trench. And there's the Challenger Deep, the deepest point. And a bunch of us got together with some physicists and a bunch of current data we gathered. It took a lot of us to put this together. But we came up with something called the trench funnel hypothesis, which is that the trenches might be different than the rest of the deep sea, which do get very little food, because they might act like a collecting funnel. And this is based on current data and all kinds of stuff, and even theoretical physics for some of our authors up here, that the trenches might not just get a rain of material from above, but they might collect things from currents from all over the place. And unfortunately, we were right, as you'll see. Okay, so our plan was to do proper transects. I skipped over a lot of history, but everybody that had done the Mariana Trench before that just went straight to the bottom. They ignored all of this. And so we were going to drop the Nereus and the landers all at points all the way down. It's called an ecological transect, and that's what we set out to do. So let me show you some of the things we found. I'll have Kermadec and Mariana, South Sandwich scattered in here. But here's the lander on the side of the Mariana Trench. This is the upper section, upper half. And this was a new shrimp species. These mysterious rings, we have no idea what those are. These mounds, we have no idea what those are. This is a crop circle made It's made by a Yoda worm, and this is a Yoda worm. This is one of my professors at Scripps had just discovered it 2 years before, and we got the second image of it, except the worm's not very good in the picture here. But they feed in crop circles. Sea lilies, crinoids, these are relatives of sea stars. This is a slime star. Has anybody ever gotten a slime star from out here? Yeah. We have these out here, but the deep sea one, the slime stars have this unusual breathing hole that the ones out in the Salish Sea have the same thing, but they're much smaller breathing holes. This is a record high for a breathing hole on a sea star, and most sea stars don't have that breathing hole. So that's a new species, we think. We thought there was this 8-arm thing sticking up. We don't know what that is. We didn't. The Nereus couldn't pull it out. Mariana Trench, we found a shrimp. We nicknamed it the fighter jet shrimp. It has a clear canopy over the thorax. And it's also clear back here too. And we have no idea why that is. Why does it need a clear thorax? We have no clue. This polychaete worm has some really vicious jaws on it. We have a really blurry video of it running at high speed across the sediment, and one of my students said she had nightmares from it. So I'm not going to show that video because it's really, really blurry, but it went into one of the traps on the Nereus, so we were able to bring it back up. A new species of stalked sea star, and then cusk eels. Cusk eels were known from all the way back to the Challenger, but never before seen in a trench. So the next thing that made all the news is our first expedition. We discovered the home of the supergiants. And if you haven't heard of supergiants, well, this is what they are. They're called supergiants because they're amphipods, and this is your normal amphipod. There's my finger. That's a beach flea, right? You've seen the beach hoppers out here. Yeah, those are amphipods related to your roly-poly isopods. These are huge. Yeah, these are record-setting. This was kind of a Bigfoot of the deep, sort of. There have been a few caught in the 1890s with nets, and then Bob Hessler got a couple of pictures of them, and they were never seen before or after or since until us. And they were out on the abyssal plain that they got those, not in the trenches. So we discovered why they couldn't find more. It's because they live in the trenches. And this is the way the media, Whitman and Fox News covered it. And they're scavengers, and they go right for your bait and on your lander. What we discovered is instead of having meat back here in the tail where a shrimp would have meat, it's full of this buoyant fluid that makes it kind of like a zeppelin or something you'll see in the video. It's high in TMAO, and it's this oily liquid fat, and it makes it float. So I'll show you this video here, and here's a supergiant on the lander bait, and here's the cusk eels trying to eat it, and see how it behaves kind of like a blimp or something. It just floats around, and then it kind of bounces around when it gets hit, and then it's got its little propellers back here. And there it goes. And the cusk eels, we're the first to ever get a video of them. They have this suction tube kind of mouth, and they're trying to eat the supergiant, but the supergiant's not having it. They have these really spiky tails, and I actually cut myself on one of these tails because it's like a hypodermic needle at the tip. So that's why they can't eat it. That's probably how they get to be so big. They're really hard to eat. So that made all the news media. And the snailfish. So the snailfish are that group I told you about. You can find even in tide pools, but these are all different species. We found them in all 3 trenches, and they were all different species. This is the Kermadec Trench that Alvin had rediscovered from the 1950s, but this is a lander we set down, and they're everywhere. They're all over the place. There should not be this, and these are amphipods, but there should no way be this many fish down there according to earlier dogmas. And they're just everywhere. And this is the new one you'll see in the video that my student Mackenzie got to name. It has sensory pores, it's blind. She discovered they have some kind of array of sensory taste and smell organs here. New species, in the South Sandwich Trench. And then the one that made all the news media is the second species we discovered that no one's ever captured. This one goes into traps easily. This one I'm going to show you, it's never gone into a trap. Nobody's caught it. I'm kind of glad they haven't caught it. Mackenzie Garinger, my student, named it the ethereal snailfish. But this is a geology lander with tube cores. And here comes this ethereal snailfish. And what's different is it has a head like a dog. It's a puppy dog. Alvin named it Snoop, and my student named it Ethereal. And what it's doing here, it's blind, we think, and it's homing in on something like a worm that we dug up with the lander by accident. And somehow it's just homing in on that. We don't know how. And it comes in and it's going to eat. That's just, I'm kind of glad nobody's caught this yet. The Chinese are trying to catch it right now, actually. And yeah, that made all the news media. This was #1 on Facebook for 3 days in a row. And it's just beautiful and diaphanous and yeah, I don't know, kind of magical. Okay, the lower zone down all from somewhere around 8,000-ish down to 11,000. No fish have ever been found at all. I have some ideas about that, but I don't have time for all that. But anyway, I'll just show you some of the things. We found sea pigs in all 3 trenches, different species. Nereus grabbing one with a suction tube, actually. Swimming sea cucumber. Here's the South Sandwich Trench with sea pigs all over. Deepest recorded jelly ever in the Mariana Trench. Here's another jelly in the Kermadec Trench. We don't know what it is. I don't know if you can see the dense tracks there, but we think those are made by these worms. And we got this thing, and we have no idea what that is. I don't know. It has these lobes on it, and we don't know what it is. I don't know. Here's another very different kind of amphipod. It looks similar to carnivorous ones, but that are shallower. So we think that's a new species. It's a very different kind of amphipod. But let's go to the amphipods now. So to finish this part up, there's no fish to eat them as far as we can tell, and the amphipods go crazy. They just seem to have no population control other than parasites and food, I guess. But you'll get thousands in one little lander trap this big, and it'll have 1,000 amphipods in it that come out of nowhere. And I showed you that one nightmare video that Alan had done before. Here's one from the South Sandwich Trench, where we're near Antarctica. That's the sub. I didn't get to go in it, but the sub was on here. And this is an amphipod before it eats, and this is after it eats. It's got, unlike most crustaceans, this one has a soft belly in it. Yeah, it can stuff in. So these amphipods are there in huge, huge numbers. This violated all the original ideas about the deep sea. There shouldn't be this much there. But the funnel hypothesis turned out to be right. So, and the other weird thing is our geneticists did the DNA and found out this is the exact same species as the one in the Mariana Trench, several thousands of miles away. It's completely unexpected. And here's one of my pictures here. The amphipod has yellow things are a naked retina, but they have these red things. They're not eyes. We don't know what they are. There's no visual structures in it. We don't know what those are for. We found they have lots and lots of interesting osmolytes, some of which fish don't have. In Japan, they discovered it reinforces its exoskeleton with aluminum that it gets out of the sediment. Very, very weird. And last but not least, our funnel hypothesis turned out to be correct. Stuff is collecting down there. Alan's lab found record high levels of PCBs and microplastics a few years ago. We analyzed them with Frank Dunovit at Whitman, found tons of mercury and lead, way more than you should eat if you're pregnant for sure. Just unbelievable amounts. The Chinese have built their own DSV now, and went down to the trench 3 or 4 years ago and found this all over the Challenger Deep, all this trash. So it's turned out to be a garbage dump, and it's pretty distressing. Okay, I need to finish up with some hydrothermal vent stuff. I just want to show you some pictures from our Alvin dives here. So here's the edge of the Rift Valley on the Juan de Fuca Ridge, right out wherever, I don't know, wherever, that way. That's the edge of the Rift Valley. As you come down the Alvin, here's a picture on the seafloor. Here's another animal attack, and here's the edge. This is pillow basalts on the edge of the Rift Valley, and those only form in the deep sea. And you can drive right up Hurricane Ridge Road and see the exact same thing. So this is ancient seafloor up here on Hurricane Ridge Road that got shoved up out of the ocean. So those are exactly this. And you can't get those on land, only in the ocean. Another couple of things here, a vent, and we'll see more in a second. So anyway, I don't have time to do the history of this, but basically Kathy Crane is the one that was responsible for their discovery. She didn't get credit until NPR decades later finally broke this to say, hey, stop giving credit to all these other people. In a nutshell, she set out to solve the marine minerals mystery, which is that half of the salts in sea salt could not be explained. I have no idea where they came from. And she knew they had to be the hydrothermal vents because the ones on land are putting out just the right minerals. But as I told you before, she was told that can't be. In a nutshell, she persisted, got some thermal data, and led the Alvin to this revolutionary discovery. And there's a link to read this if you want at the end. And that's the hydrothermal vents. And this was not just a geological revolution, it was a biological revolution because there were no biologists on that expedition. She's a geologist. Tubeworms, giant tubeworms up to 10 feet tall, living around the vents. The vents themselves are making huge chimneys. The water was below boiling because of the pressure, so it could get up to 750 degrees. And all these animals were completely unexpected. So giant tubeworms, giant clams, giant mussels. This is what I saw, but I'm using Ocean Networks Canada's videos for these because their cameras are much better now than Alvin had in 2008. But I worked on all these things. So we have a smaller tubeworm that was named Rigia. This was named Riftia, really original names, right? Riftia on the ridge, Rigia. So we have them too. They're just a bit smaller. But they're all over out there, and I've worked on lots of them, and I have some in a freezer. It's still at Whitman, but I'll have to bring it out here someday. Yeah, and they rumble. This is another one. This is exactly what I saw from the Alvin, but Ocean Networks Canada, so like I said, their videos are a lot better now with 4K cameras that we didn't have. So this, and this is in Canadian, it's just over the Canadian border off of our coast. And some of these chimneys are ridiculous. I mean, this is one I saw. I have almost identical video. It's just blurrier, but this one goes up and up for like, I don't know, more than 100 feet tall. Just up and up and up. And the geological revolution, as I told you before, they've been found all over the world, and they completely solved the marine minerals mystery. They put out minerals that are vital to life. Life would not exist without these minerals, and we want them, or people want them, right? And some of these are just ridiculously extravagant. These were just discovered, the pagoda vents by Schmidt, the Google ship. Mariana's Rift is a spreading center away from the trench.
Oh, this is so cool.
That's this.
What, 27 meters high? 30 meters high?
Anyway, whole bunch discovered with AUV by Monterey Bay Aquarium Research Institute right off our coast just a few years ago, hundreds. Oh, I should go back one click. The Arctic, unfortunately, is opening up from sea ice melt, so now they've discovered them here. They're in the Arctic. And then these are loaded with minerals, a different mix of minerals than the polymetallic nodules, but still lots of minerals that industry wants. And what's happening here very quickly, basically, is that seawater percolates down in here, gets heated up, dissolves all of the Earth's bounty. All the minerals get dissolved, well, not all of them, but lots of them. Gets heated up, comes shooting out at up to 750 degrees, and then when it hits the cold seawater, a lot of it precipitates as these chimneys, and I have a baby chimney up here. We had to get a permit from Canada to collect it, but they let us collect it, so it has a permit. But anyway, yeah, all these minerals precipitate, and then the rest of the water goes out and fertilizes the oceans with all these minerals that life has to have. And it's full of rare earths. And anyway, mining companies want to do this. This is the tractor that Japan has built just last year. So this is going to go down and suck up nodules and vents. And, smartphones, pixels, you need all these rare earths and so on. Norway was the first country actually to start, but they got enough protests that they just last month or 2 months ago put a hold on it, but who knows for how long. So this is going to happen. I'm kind of out of time, so just in terms of the biology, just in a nutshell, this was a revolution in biology because these turned out to be running on chemosynthesis instead of sunlight. And what happens is the hydrogen sulfide gives energy to microbes, and the microbes oxidize it and are able to make sugars and amino acids just like a plant would. And they're symbiotes. So this little Regia that I dissected here, instead of a gut, it has a sac of symbiotic bacteria, and it feeds its bacteria with the right gases. The plume picks it up and feeds them, and the bacteria feeds the host with sugars and amino acids. And here's the food chain, another attack on Alvin. All these things going on. And you get huge quantities because of all the energy down there. And last but not least, the other vents in the world don't have tubeworms. In the Atlantic, it's shrimp in huge quantities. On the Pacific Antarctic ridge, it's crabs with all the symbionts. Anyway, it's a huge amount. So I don't have time to tell you about our work. We've found a bunch of osmolytes and things on them, and so I'm kind of out of time. But and I don't have time to do the seeps and the seamounts. I didn't think I would have time, but you should be aware that seamounts have ancient species like 1,000-year-old coral gardens, and they're being trawled now. And the methane seeps are areas with huge amounts of frozen natural gas, and yeah, those are going to be extracted. So that's all another whole talk in itself. So I'm going to end there, except you should be aware China's about to do it, Japan's about to do it, and I'll sign off. So, but basically, this is just to read during the questions. So thank you to everybody. This is all medical applications that came out of TMAO that you can look at on YouTube later if you want. The Hades group, some links, and a summary, take homes.
Thank you, Paul.